afternoon, everybody. Today, we're very honored to welcome Professor Ahmed Abdelaziz, a leading mycologist and a food science professor at Sultan University. With a focus on the biodiversity conservation and ecology, he has significantly contributed to the field. In addition, as the founder and the president of the Earth Society for the Congo Conservation, his expertise has shaped a global research. We're very excited to have him here with us today, and we're looking forward to his insights. In addition, we're delighted to welcome Dr. Iasmina Moza, an accomplished physician and an assistant professor at the University of Bucharest. We're happy to tell you that she has done her PhD on the aquatic ecology. In addition, she's pursuing a second PhD on the molecular degrading cultural heritage. We have also mentioned that she is the founder of Moza Foundation and she is the president of Rosa, which is an international company called Nile. We're very excited and very honored to have you with us today, and we're looking forward to your insights. I hope you all have a good day and take as much of information as you can. Thank you.
Hello, everybody. So I would like to talk to all of you by Franco-Arab, if you like to. My name is Ahmed Abdelaziz, or you can say Zimo. Okay, so it's Zimo. I am a professor of mycology in Sultan University, and also I am the founder of Arab Society of Fungal Conservation, along with the president of African Mycological Association. So it's my honor to be here with all of you and with my friend Professor Sana and Dr. Mariam. It's my honor to be here for the first time in my university. And my work is related to fungi, Patria. So all of you, some of you studied microbiology, and the others studied agricultural microbiology, and the rest studied environmental microbiology. So what is the role of fungi in our life? We know that fungi already considered the most important organisms in our ecosystem, because it's considered the biotic component, because its ecosystem is composed of abiotic component and the biotic component. So fungi considered one of the most important biotic components in whole ecosystem. Why? Because it's considered so fungi considered the most important organism among biotic components of the ecosystem, because it's already released huge amount of enzyme to degrade the most release the amount of byproduct, which is called lignin. So fungi are the only organism all over the world able to degrade lignin. So fungi considered the core of the ecosystem, which release huge amount of enzymes, able to biosorb heavy metal, able to produce drugs and medicine, able to eat our cultural heritage. So I am lucky to work with Professor Zay Hawass in the Chibo's second ceremonial booth, and with the American team for the Abydos cultural heritage. So fungi able to eat our cultural heritage all the time. Also, fungi producing huge amount of agricultural applications. Looks like plant growth promoting criteria, looks like biostimulants. So different applications of fungi. You can read it in my books. I have already published about 17 books worldwide. And also, one of the important things related to fungi, the recent application in biofuel, batteries, a lot of application of fungi. So today, it's my honor to present my colleague Iasmina Moza. Sure, she know what is meant by Moza, and she laughing a lot. No problem for that. Her family name is Moza. So Iasmina is working for a University of Bucharest, and Iasmina working right now to obtain her second PhD, because she already got her first PhD on Cyanobacteria in Danube. Yeah, and she still survived, sure, no problem. And she's got her PhD second one, because Iasmina is working on fungi deteriorating cultural heritage, Romanian cultural heritage, wood special one, and she is working on that field. Soon she will get her second PhD. So welcome, Iasmina, to our presentation today.
You can. Okay. Thank you all. I'm very nervous and very honored to be here for my first time to give a presentation in Egypt. I was here 30 years before, and I was very little, so I don't remember very much. So now, yeah, I will talk to you today about the Danube Delta Biosphere Reserve, because also we have in Europe our Nile, let's say like that, is a river called Danube. And in my country, this river form a delta, which is a hotspot of biodiversity. So it's very important to be studied, and of course to highlight about climate change, especially toxic cyanobacteria that we have there and all over the world nowadays. As Rimo said, I'm teaching at the University of Bucharest, and I'm working at Faculty of Biology, Department of Botanical and Microbiology, so the same as professor. So I will just give you a few things about, very quickly, about my university, because we have a bit of different programs. So here you can see a picture from the capital of Romania. It's Bucharest. And can I use this? Okay, so here is one of the main buildings of the, thank you, of the university. And about the faculty that we have, I will not give you too many details, but you can see the numbers here. I don't know how many students you have here, but we have 90 faculties, including faculty of biology, mostly everything except the medical faculty. And of course, we have different doctoral school of various fields. And recently we have also an interdisciplinary doctoral school. And we have mostly 34,000 students, and of course, a high number of the researchers, because we also have one research institute that belongs to our university, where we also have programs for postdoc students and also for visiting professors like Zimo this year. So I also want to highlight that for bachelor studies, we have 3 or 4 years, depending on the faculty. And for master students who have 1 or 2, we don't have pre-master like you have. So this is a difference. And most of the PhD studies are 3 or 4 years. Of course, we can extend in some situation. We also have a nice botanical garden where we can do our labs outside, and mostly here we have also, you can see the numbers, a lot of plants, and we can do experimental fields and so on. So we are lucky that in the middle of a very crowded capital, we have also a botanical garden. So in order to take you now to Danube Delta, I wanted to specify that we also have some important facility belonging to our university that are based along the Danube River and the Danube Delta. So you can see here the four of them when the Danube enter in Romania, and then in the end when the Danube enter in the Black Sea. So mainly my research area was here on the Danube Delta. So I will talk more about this. In order to give you context, I think most of you know what are cyanobacteria, yes or no? Of course, you are very smart, all of you, yeah? So I will not give too many details, but you know that they are often called blue-green algae, but the correct name is to say Cyanobacteria. Blue-green algae is given by the color, so you can see here a picture, how they look like a painting on the surface of a lake, yeah? So of course they can produce photosynthesis. They are very specialized microorganisms. They are also similar with fungi in terms of specialization. And of course they pose morphological and ecological diversity. Yeah, we can find them everywhere. And what is the negative fact about them? Besides they are primary photosynthesis, they can become toxic, yeah, deadly. So that gives a ecological negative impact. So I study more deeply in that. So you can see here the Europe and Romania highlighted with red, yeah, and here the localization of Danube Delta. I took again this map because you can see on the Danube Delta area four different colors. Danube Delta is split it in four lake complexes. Like, for example, those three are called fluviatile lake complexes. And this one, here we have a sandband. It's closer to Black Sea. It's called maritime lake complex. So why is this important for you to know? It's because we wanted to find out in this study case what makes the cyanobacterial bloom toxic, because in the at least last 10 years we experienced a lot of toxic bloom, and I think you during the summer here, but the temperature is much higher, you could see that. So we know from the literature that even if we have a small amount of rain, let's call them like that, in a water body that with red are toxic and mostly are not toxic, we can have a shift in community composition and most of them to become toxic. And also we need to follow all the environmental driver in order to know what they make them to be toxic, and of course to prevent that, because we are talking about fresh water, we are talking about water that has a very meaningful for the agricultural purposes, yeah. And also from the literature we know that if you put a salad on the ground that is water with toxic cyanobacteria, that can be accumulate and we eat it. So this is very important to be studied and to be stopped. So therefore we were interested to see if our cyanobacteria from Danube Delta shallow lake, they are called shallow and I will explain why, they pose toxic genes. We wanted to see that. What kind of toxins, because there are more, I will tell you more things about that also. Of course, if we can talk about a health hazard in the Danube Delta, and we were also interested because we had so many lakes in Danube Delta that are under different environmental conditions to search for the main drivers for bloom. So we followed this question, yeah, and mostly were interested to see the ecosystem consequence in the case of cyanobacterial bloom. Because till now we didn't have like a real bloom. Like for example, I saw in Greece some years ago, we have localized bloom like a skunk, and that's it. are lost. Therefore, I will give you this outline, and in the conclusion, I will rename them and we'll give the conclusion based on them. We selected 26 shallow lakes from Danube Delta in order to cover all the lake complexes because they are different. Some are older, some are new, and so on. And I need to highlight that it was the first time when someone did molecular investigation in this area. Till then, only a classic taxonomy under the microscope. Yeah, of course, we didn't have these climate change problems at that time either. So, of course, we are interested to see about the types, also occurrence and distribution during time of these gene markers that we follow. So why we choose for this Danube Delta shallow lakes? Because, as I told you, it's a unique biosphere reserve and it's very close to our university, and we have a research station there. We have more than 400 freshwater body in this area, mostly shallow lakes, yeah, 4 different lake complexes, so we can group also our analysis during statistics. And of course, we have high phytoplankton biodiversity, and most of all, because that's why I decided to do my PhD, that lack of data regarding cyanotoxin, and how I used to say, because it's simply the same but different each time. So here you can see a channel from the Danube Delta. It's an original picture. So in order for you to understand about Danube Delta lake heterogeneity, and maybe it's the case also in Nile or other delta, but it's more highlighted here in Danube Delta. It's the fact you can see here some original picture. As I told you, they are called shallow lakes because the water level vary between 0.0 meter and 4 meter maximum, depending on the rain season, of course. Yeah, and you can see some pictures. They are very clear. Even I could show snails underwater, you know, walking. I also did this type of diagram for you to be more safe explaining. Yeah. So in Danube Delta, it's very important the connectivity for play. In Danube Delta, when Danube reaches Danube Delta, it's spread in 3 main branches. One is very close with Ukraine, by the way. So we are a bit at risk. We are not allowed to go there to sample. Yeah, we can hear the bomb and everything. And one was already, yeah, it's used for navigation, and it became dry. And there is also the third one. Between them, there are many lakes. So once you go forward from the main channel, you have different type of lakes. So I tried to make here to see that we have either sandy bottom, clay bottom, organic bottom. We have a lot of reed beds. This is specific for Danube Delta, and they are floating. Yeah, they are not still. So we also experience a lot of bad situation when we go to sample. What is also important that the scientists group the lakes beside the lakes complexes in 3 different lake types based on how often they bloom per year, yeah, and based on phytoplankton diversity. And we started from this because we have some data, and we wanted to see if, for example, in one lake with, let's say, organic bottom and higher blooms, we'll have also toxic cyanobacteria or not. Yeah, so we started for this. Here, just to give an example for you, I choose from the same lake complexes. Yeah, so here, like for example, we can experience like that are with a larger surface, but either with good connectivity or with poor connectivity. So it's not a rule. The fact is that with Danube Delta, everything is not a rule. What I found about other cyanobacteria and shallow lakes in the literature. In Danube Delta, it's not the same. It's either the other way around. So I could not really use the information that I found in Danube Delta. We also have a seasonal dynamic. So here again, I try to make some diagrams for you to be very easy to understand. So this part represents the sediment, as in all these pictures. Those are original, yeah, sediment and water. And here you can see the water with blue. So this is a typical state for any shallow lake. Yeah, so you can see there some picture. When we have 0 or less sediment suspension, yeah, what happened in Danube Delta? During flood periods, where it's usually in April, May, yeah? So we have high water level, of course. We have high sediment resuscitation rate, so you can see the water, yeah? And with this diagram, I try to mix the sediment with the water. What happened then after flooding period in Danube Delta in July, for example, yeah? Depend on the lake type. As I told you, there are 3 different types with different types of sediment. Yeah, so depending on that, we have different situations. But all the time we don't have sediment resuscitation because we don't have floodings anymore and the water start to lower, yeah, the level. So you can see, like for example, in another type of lake, yeah. And we also experience this extreme, very extreme situation because just imagine if you have one lake that is during the flooding 0.5 meter, yeah. In August, when we have not 50 degree, but 35 or something, 40 maximum, it will still survive. No more water, so it's completely dry, yeah. And in September, the raining again start, and we have a mix of organic matter, mostly came for plants that are very well development during the summer. We also tried to, for you to understand, because I mentioned about different sediments, I just made a quick graph and diagram of how many types of sediments and combination we can find. Yeah, so there you can have, based on those 26 lakes that I study, I will not tell you the name because you don't know the Danube Delta, but you can see that more or less they are basically grouped. Yeah. Okay, so now I would like to give you a few scenarios about toxic cyanobacteria, because you know what are cyanobacteria, but we don't know how can they become toxic. So again, I try to explain in a very simple way. So we can have or not the bad species, let's call them like that, yeah? So we can see here green algae, yeah? In one situation without cyanobacteria, and where toxic TP means toxic toxic potential, yeah, it's present, yeah, or Iasmina. In this situation, I try to make an Iasmina, yeah. So when we have the bad species, either we can have the bad gene or not, yeah. So you experience 2 different situations. You can see the gene highlighted there. Okay, next scenario. Okay, we can have the gene, we can find it, yeah, using genetic markers, but we don't know if the gene is expressed or not. So for that, we need to go much deeper and to find some environmental driver. Even more, to get more complicated, yeah, to have more fun. If the gene is expressed or not, they can produce toxin or they cannot produce toxin, yeah? And the amount I try to find it, yeah, can be different. Again, in strong relation with the environment, we can have low, for example, microchystin concentration, which is the hepatotoxin, yeah, low or higher. So if you want to, for example, search in the water and not in the biomass for the genetic method markers, and want to quantify the toxin to see if it's low or high. We don't know if that cell released by themselves the toxin, or it's because due to the death of the cell, basically cell lysis. So there are a lot of questions. Yeah, and of course we also experience a very hard situation when we try to do also qPCR, because due to the diversity of the segment types, we had a lot of inhibitors for our PCR reaction. So we needed to take this into account. And of course, we could have, like, for example, in the water, we'll have a stronger qPCR results because the gene were unnaturated and so on. Yeah. So this was like, I think now everyone understands what is with cyanobacteria, not only in a shallow lake, but in general, in any water ecosystem. So of course we did some in situ measurements. So here I just put some pictures to see how many things we took with us, not because I'm a girl, because we need it. Yeah. So also in our small boat. Yeah, and we had. I started with this very old. How, I don't know how you call it, but we call it chalupa. Yeah, a very small one. So I also had a lot of fun because during the low water level, we had a situation where we need to carry, the small boat, because the entrance of the lake looks like that. So you just simply need to carry. Yeah, so a lot of fun. We also did, we had this very nice device. I don't know if you have it or you know it. It's called a fluorometer. So you put it in the water, and with a laptop or a USB stick, it gives you this type of graph. It's a very expensive one, but it's very useful because it can give you live measurement about green algae, blue-green diatoms, cryptophytes, and yellow substance. Yeah, so this is very important for us to know if we have a bloom there or not. Yeah, and of course you can also count cell with this. It's a very good device. And of course we measure a lot of things in order to have like the depth, the water temperature, turbidity, transparency, oxygen saturation, and we can see, like I choose here, 4 different lakes from the same lake complexes. Like for example, this one, with this one, they are very close to each other. So it's clear that there are strong local environmental conditions for those lakes, which is make my job harder, but from the scientific point of view, it was something very extraordinary. And of course, we wanted to see about the taxonomy, and here there are some pictures, so you can see the high diversity of cyanobacteria that we had in our lakes. And of course, mostly we were interested to do some genetics. So basically, we did filtration in each lakes, and we preserve it with alcohol. And of course, we follow this preliminary general step. I will not give you too much details, but what was important for us that we use, because cyanobacteria have a lot of exopolysaccharides, so it's very hard to do the DNA extraction. Yeah, so we tried the different kind of commercial kit, but in the end, we decided to optimize our own method. It's also published here. I just put a small scheme. It's based on potassium ethyl pentogenic. Yeah. So of course, we were interested mainly to search for our markers. Yeah, to see if we have and what type of toxin we have. So here you can see different words that are related to different type of toxin. Yeah, and basically we did DNA extraction and PCR amplification plus qPCR, and then we send it externally for pregnancies and so on. And like any other research, you need the first step to, read a lot of literature, and we selected based on our taxonomy results, because I want to go narrow, yeah, the primers from the literature, and we ordered it and we used that. Of course, we did also some statistics. I will not enter into much detail, but I work with two different statisticians for more than 4 years in order to obtain some graph that will short I will present you here. So it was very, very hard. Yeah. So let's see about the diversity and abundance. And I just put one small picture with pie for you to see. With red, we have cyanobacteria. Yeah. Based on that apparatus device, fluorometer. Yeah. So with that, we can obtain in 2 minutes this kind of pie. So we'll know for each season, May, July, September, because I didn't mention, but we choose to try every year, not winter, because it's very dangerous to go there, yeah, but to see the seasonal dynamic, yeah. And in terms of abundance, total abundance expressed as cell per meal, we saw, like, for example, Iasmina and microchips, two very potential toxic genera, yeah. We could see that according to World Health Organization, they cross the level and they present, those water bodies present moderate health risk if they are toxic. Yeah, and based on the analysis that we have from our taxonomies, we highlight few genera that could present toxin. They could produce toxins. So you can see there are few. Yeah, and also when we wanted to see the main cyanobacteria with toxic potential at dominance of the genera, yeah, we can see that we have mostly Oscillatoria, microcystic, Lingvia, and Iasmina, except microcystic or are filamentous, yeah. So they can travel with the ships, they can resist to high waves and so on, because we have a lot of storms. And also microcystic have the capacity to have air vesicles and travel on the water column to reach sunlight, to do whatever she wants. So that species is very, very dangerous. So now the hard statistics that I will try to make it simple for you. Yeah. So where you see S or O, it's from September or October. Yeah. M from May and G from July. Yeah. So we wanted to see if we can have like a pattern. Yeah. Like to have more genera and higher amount in which season. So you can see here, for example, that they are concentrated here next to this triangle, yeah? And also there are two different years, yeah? 13 and 14, there are two different years. So in first year that when we study, we have a higher amount of toxic cyanobacteria during this year and during autumn, because it was a very hot autumn that year. So has a logical explanation. In May, we can see only few genera. Yeah, so you can see here some of them, like for example, Lignia and Trichoderm. There are mostly in July. They give bloom in July. So in some lakes we have bloom in summer. In some lakes we don't have in summer. I know that is strange. I told you no rules with Danube Delta, but we have in autumn. Yeah. So here maybe you can see better that when we put all the data together, yeah, we have here most of this line concentrating on the summer and autumn. And spring is a bit far away. That means here we have higher abundance. Yeah. So if you go forward, we have lower abundance, and here you can see the same situation. In spring, you don't have any triangle with genera, so everything is concentrated here, summer and autumn. Yeah. So this is what statistics is doing to show us all the big data in a very simple graph, so we can discuss it. Because I showed you that we have 4 different lake complexes. Yeah, we wanted to see if there exists a geographical pattern, because Danube Delta goes like that. So this is according to the GPS point. Yeah, they are not put it there only like that. So there are some points. Yeah, each coin meaning one sampling point. Yeah, so we have 6 because we sample 6 times. Yeah. So we can see where we have blue, more blue, it means we have more genera prevalence, let's say like that. So you see here, we have with blue. So the Danube go like that, and here it's a black sea. So everything is basically somehow concentrated in that area. And when we did the same thing, but in terms of sulfur meal, like total abundance, a bigger circle mean more sulfur meal. Yeah, so you can see here, most of them are bigger circle than the others. So again, statistics, and of course I put purple because I like purple. Yeah, so I am sorry. Yeah, so what I told you about, now we can see in terms of numbers of records. Yeah, and here are the name of the lake, but I will not refer to them. I will just make some row for you to see how the after flooding, how the water levels go down. So it's this, and everything is concentrated in this area, in this part called maritime delta, let's say like that. So it's clear that something happened there. It's something interesting there. So of course we try to figure out and to find out more about environmental drivers. So of course we did a lot of chemistry for soil, water, and so on. And what we wanted to see, because I told you in the first slide that they present high ecological and morphological diversity, also cyanobacteria. Yeah, so I tried to put here a few pictures in order for you to see how different they are. Some are single cells, some are trichoma, some are yeah, like for example this Merismopedia. Yeah, so what we observed after we did the statistics based on all the data, that, for example, if you follow the rows, yeah, the bigger one is pH, yeah. So you can see there limbia, yeah, with the triangle very close and in the same direction with the area, means that she's sensitive to the pH, or she reacts to some pH. And once we have this result, we go in the raw data in the table to see the number, the exact concentration, the pH, if it's 7 or if it's 9, yeah. So here we see where is microcystis, yeah. So here, for example, we have microcystis and we have oxygen saturation and oxygen concentration, yeah. So she's directly affected by oxygen concentration, but on the other way around, you see the light. She's not affected by light because I told you she can travel on the water column and get how much light she wants. So she doesn't care about light. But in contrast, for example, Plantotrix or Aquanocaps or Anabina, they need light, yeah? And also you can see their nitrogen form and so on. Some of them are sensitive with conductivity. So it's basically the first time when someone did such an extensive study and to discuss in this way about this cyanobacteria. And more, because we saw that we study 2 different years, and we obtain, I put here Anabina, yeah, which is the filament. I showed you when I made those scenario, yeah, I constructed based on Anabina. So she's the only one that changed her places when we analyze the 2 years separately, yeah, 3 seasons in 1 year, 3 seasons another year. So that made me think, because Anabina can produce 4 types of toxins. So this is very dangerous. So what we were interested to see, and then I extract from an archive the temperature at Romanian level. So we see that, for example, in 2013, when I did those first studies, I'm still now working on some prediction model based on this and what we did after that. Yeah, so I put May, July, and September. So we can see here that we had a very hot summer and a very hot also autumn, yeah, with a small amount of rain. But in contrast with the other year, we had a lot of rain, yeah. So you can see the temperature and the rain level. I will go back one more, yeah. So I observed that in this situation, Anabena, who is the filament, can travel, yeah, can be washed during the water level. Thank you. Can be washed, and then she reached that point, and she we have more genera concentrated and cell concentrated there. So again, we apply another test. It's called PCI. Yeah. So we can see I highlighted those lakes from that lake complex very close to Danube Delta, to Black Sea, sorry, with red. Yeah. So I could see that this is in the first year when you have less rain, it's a bit more concentrated. Yeah. Green is on side, red is on the other side. When you have a lot of rain, and Delta became almost like a huge lake. Yeah, they are very well interconnected. Yeah, the lakes, small lakes are not isolated anymore. You can see that we have red everywhere, you know, among other points with green. So this is something very interesting, because we can talk about the concentration during the water level retreat, yeah, in a part of Danube Delta, yeah. So when we did the genetics, of course, we extract the DNA and we compare, and we see this. It was very interesting because already when we measure with nanodrop or with qubit, simple measurement to see how much DNA, total genomic DNA you have in your samples, I highlighted with 3 different colors based on the season. So you can see already from the DNA that we have different columns, yeah? They are, some are smaller, some are, yeah? So for example, with red in September, when we had blue, of course we have more DNA. So everything, it was correctly done so far. And of course, we also, when we search for 60S, we obtained very nice, I call them fat bands of DNA, because it was very hard. We need to optimize in Switzerland and in my country also. Yeah. So after that, we start to screening for those toxic markers. Yeah. So we screen from different type of gene. For microchistine that I told you, they are hepatotoxin. We also screening for saxitoxin, which is a neurotoxin, attacks your brain and you are paralyzed, and it's mostly found in peridium fishes, like in the sea and ocean. So since that those lakes are very close to the sea, yeah, Black Sea, it was very easily to, you know, gene transfer, lateral gene transfer. Yeah. So what we found, I put there, we tested in Cluj, and then we tested also in Switzerland for validation. Yeah. So we found positive here. This is the letter. We found positive, yeah, for saxitoxin. We also test anabatoxin and oscillatorin. We didn't find it. We tested also different gene for microcystin, and we found only this, which is like a general marker. Yeah, so it was clear that our algae, cyanobacteria produce toxins, so it started to be alarming. And then I will just make dynamics of the gene presence. Yeah, we have microcystin with blue and with saxitoxin with red, and I will just press the button for each season for you to see the dynamic, how diverse it was, and how hard for us to find the drivers and to answer our question. And of course, I will come again to the outline. Yeah, we study more than 20 lakes in Danube Delta, and we found that most common genera that was present there was Iasmina, of course with different species, but also Aphanizomenon, and we have the highest record of toxic potential genera to the complex, which was much closer to the sea. And of course, we had a lot of microcystis, but it was not present in all the lake. And we can also talk about double or triple blooms. What that means? We have the same amount of cells per liter given by different species, yeah, either microcystis plus oscillatoria plus annabella. So a soup, a mix that can give you a lot of toxins, yeah, for everything to be more fun. And I told you that it was the first time when we did this kind of molecular screening, yeah, for phytoplankton. And it was hard because we didn't have any previous DNA data for comparison, so we don't know. We also try to extract historical DNA for sediment core, yeah. And I will not give you the number of samples, but they were a lot. So based on eDNA, we found 68 in most of them, yeah. And of course, you could see the dynamics also in terms of DNA concentration. And of course, our genetic data fit until now in 90% with taxonomic data. That made my colleague very, very happy and a bit arrogant indeed, but it was okay because she was very happy to find out that she knows how to identify, the species. Yeah. So in terms of types and occurrence, and of course distribution of gene involved in cyanotoxin synthesis, you could see already from those very easy map that we found 3 different type of genes, yeah, 2 belonging or represented markers for microcystins and saxitoxin. When we find saxitoxin, I need to publish that it's during publication now. The Switzerland, the guy were very amazed, and they made me repeat the analysis 2 or 3 times to be sure. And I did that, no problem, yeah? So, and of course, in most of the samples that we find saxitoxin were related again to that lake complex is much closer to the Black Sea. So, in order to finalize, are toxic cyanobacteria and Danube Delta shallow lakes? Yes, we saw that there are several genera that present toxic potential. What type of toxin they could produce based on molecular markers? And I show you, and I told you about saxitoxin and microcystins, yeah, and you can see there the name of the markers that you can find also in the literature. Which are the drivers of cyanotoxin production? We don't have a strict answer yet because most likely there are a combination of factors. So we all need to have long-term data, yeah, and also extreme situation that will help us to find the exact driver and what it will be the ecosystem consequence in a case of cyanobacteria bloom, because that is the main point that we are interested in the end to do also apply research, not only fundamental, yeah? So we can talk mainly about biological impacts due to the potential toxic effect, and I gave you one. They can be accumulated in fish, shells, snails, birds, plants, if you use the water to, yeah. And for example, those toxins, if you boil the water, they will not degrade, yeah. So we will die if we drink the water, yeah. So we need to use special filters, UV lights, and so on. It's very, very hard. So just imagine that your Nile once will be all blue and you cannot use the water anymore. Yeah, you don't have that huge filter to get rid of the biomass and then somehow to destroy the toxin released in the water. So it's very, very dangerous. And of course, accumulation of organic material in like sediments increase bacterial activity. And we also test some bacteria, and we saw that, for example, they like to stick on the exopolar saccharide mass of microchips. We found a lot of pseudomonas. So you can increase also pathogenic bacteria due to the bloom of those cyanobacteria. Yeah. So community level effects on cyanobacteria blooms are not yet well understood on the international level. Yeah, and in particular in this case, the impact of toxin consists in change in food web structure and function. Mostly, they bioaccumulate in Daphnia, zooplankton, that fish try to eat. Yeah, so it led to serious ecological consequence. And of course, a lot of people eat fish in Danube Delta, so we need to pay attention on this. So of course, this area require additional research, so I'm not stopping studying with them. Even I have two twins baby, cyanobacteria and fungi. I try to, to treat them the same. And of course, I need to thank to a lot of people that were working in obtaining this data and do this project. And I also want to thank you all for your attention and yeah, for your invitation here. And do not forget to follow your dream. Yeah, this is a very important mention. And if you have questions, you can see scientific method, yeah, here. Very easy explained, yeah. Thank you so much for Iasmina. You're welcome. Who wants to study cyanobacteria from now on? You have a question?
Yes. Mina, we already studied cyanobacterial toxin in Nile Delta, not Nile Delta, but it's in Bucharest project. It's already a big issue in Fayoum, Giza, Bucharest, Kuwait. So that started in 1990 with Cyanobacteria brevis, which already making a bloom. And you know, it's already hepatic, hepatic toxin.
Yeah.
And they started that in the they started blooming between April and May. So it's a big issue in Bucharest, and I think they started to cover the freshwater channel in Bucharest, so they try to prevent the life. Even I know Cyanobacteria is a kind of trichome and lignia also.
And she likes wind, water,
Yes,
and rain.
yes,
So
and
that's
they are
why she
keeping
likes
here.
wind.
Yes, they are keeping clean the channel each year, and they damage the trichomes and they distribute the toxins in the water. And the people try to manage that by using the water filter. Sometimes it's give good results. Sometimes the poor people boil the water for a long time. It may be 30 minutes to boil water to save their life. Because I'm sure it is not the only way. But right now it's completely decreased because President Sisi is control that issue related to Lake Mandela. You visited Lake Mandela to
And you saw nitrogen input. So if you pollute the water, you increase the amount of cell per meal for those fishes.
So
also.
if to study cyanobacteria, you can come to Bucharest or Lake Mandela. Last year, I recorded microcystis, huge amount of microcystis cover the Lake Mandela and already infected fishes there. So if any great guy can come to the house and to collect sample, it will be great.
Who is planning on joining the environmental advisory plan? So you will most probably be studying topics related to cyanobacteria and toxic potential of different microorganisms that exist in the water.
So if you have any curiosity to ask me also related with my university, my subjects, me.
I have a question regarding
Yes.
this. So how did you take the decision to do a second PhD?
Because my first love was to study fungi for cultural heritage. I did that for my bachelor thesis. And after that, I took a master, a double master, to also study conservation and degradation and conservation of polychrome wood, to learn more about that cultural heritage, because we have a lot of wood churches. And then I fell in love of this subject. At that time, there were only 2 or 3 specialists in my country. And I couldn't find any supervisor that could lead me to that subject. So I took the decision. I like to go in the field. I like to swim. I will take this project, as it was with Switzerland, and I will learn about cyanobacteria because they also can be found as a biofilm on the cultural heritage. I will learn molecular technique that will help me also with fungi. I will learn microbiology. So based on that, that was my decision. After I finished this, in the meantime, actually, one of my professor, which is actually the vice rector of research of the university, started the first project on bioarchaeosciences. And they also started to do microbiological investigation on cultural heritage. And when I heard that, I went to her and asked her, please take me for my second PhD because I want to study that. In the meantime, I am one of few, I think 4 or 5 biology experts that do reports every time when some cultural heritage belong to the UNESCO needs to be restored, because you are not allowed to do that if you don't have strict, condition and rules. So I am one of that. I still doing that since 2012 now. So yeah, it was my chance to have a lot of samples. Yeah, so that's why I decided to go to the second one, because I knew that I have experience. I still have the nerves, So I say, let's do it. Yeah. So the first one is in ecology, and the second one will be in biology. So yeah, that was the that was the reason. No other reason. I don't need it the second one just for me. So, yeah, for my patient. Yeah.
So we learned from this that even if you get one PhD, if you want to learn more about something, you can go ahead and do a PhD. I think this is a very great decision.
Thank you.
So I am a neuroscientist by training. So for me, this is a lot of new information. For me, cyanobacteria is spirulina.
Yeah, okay. But we have a cytotoxin that attacks the brain, so you can go.
Yeah, maybe.
Yeah.
We recorded that and used it in the public in 2001. So yeah.
And also in the Bible, when they say the Nile became red, it was also due to some algae or cyanobacteria. Yeah, dinoflagellate that usually produce this cytotoxin. So it's very possible that toxin, killed the people. Yeah, so there is a scientific explanation for that. Yeah.
And also, she is working right now on fungi. So any guy interested also to join Iasmina or my school in Cyanobacteria University, we are working on fungi a lot. So,
and thanks to my second PhD, I met Professor Zimo. So I'm here now. So this is a very good reason also. Yeah.
Yeah, that's great. Thank you
I
so much.
also
If
thank
anyone has any
Any
questions?
question? Any question for fungi? Okay, if you like to join our team in Tuscany University, please check fungiofegypt.com. You can find a lot of information on that website. We already celebrated each year Fungus National Day of Egypt. I founded it in 2016, and this is why Akfor God is carrying a full basket of mushroom. So each year in 20 of February, we celebrated it. So you can join us on next year, inshallah. This year we celebrated it in Medical Syndicate in Cairo. Maybe next year we will be in Romania for my visit, yes. And also we founded African Fungus Day at 22nd of May. So just check fungiofegypt.com. You will find all information, all teams, all awards. So please join us in South Carolina University or Bucharest University, because if you finish your master, they offer you a PhD scholarship. You can visit Bucharest for 3 years, and you can work there, and you can be European. Okay? Okay. Okay, Professor Maria.
Thank you so much.
Ah, okay, nice. Thank you. Thank you.
Cannot, I don't know if you see me. Thank you very much, Iasmina. I like your presentation. Congratulations. I cannot see you.
Antonia. So to PhD, to flex.